For a single-celled organism like an amoeba, survival is a matter of simple diffusion. Because it is microscopic, it possesses an exceptionally high surface area to volume ratio. Oxygen from the surrounding water easily diffuses across the cell membrane and reaches the center of the organism almost instantly. But as life scales up to the size of a highly active mammal, that geometry collapses. As biological volume increases, the surface area to volume ratio plummets. Simple diffusion becomes mathematically inadequate. Oxygen moves too slowly over long distances. If a mammal relied on diffusion alone, the cells deep within its tissues would suffocate, long before a single oxygen molecule could drift inward from the skin. To bypass the physical limits of diffusion, large organisms engineer a mass transport system, a method to physically pump dissolved resources directly to the cellular level. The oxygen transport vehicle is hemoglobin, a protein with 4 polypeptide chains around central iron groups, carrying 4 oxygen molecules. Binding the first requires high oxygen concentration, triggering cooperative binding. The protein alters its shape, allowing the remaining 3 molecules to snap into place. Graphing hemoglobin saturation against environmental oxygen partial pressure creates a distinct S-shaped curve. The top right flattens. In high oxygen lungs, hemoglobin reaches near full saturation. But in respiring tissues with lower oxygen, we hit the steep section. The protein's affinity drops, rapidly unloading its oxygen. Hemoglobin acts as an active, responsive machine that holds tight to oxygen in the lungs and drops its payload exactly where cellular demand is at its peak. Distributing this payload requires dedicated infrastructure. Mammals utilize a double circulatory system, which physically separates the flow into 2 circuits, a low-pressure pulmonary route to the lungs and a high-pressure systemic route to the body. Fish operate on a single circulatory system, pumping blood through their gills and directly to their organs. But squeezing fluid through the microscopic capillaries of the gills bleeds off almost all of the hydraulic pressure. Mammals avoid this by returning the oxygenated blood to the heart, repressurizing it completely before dispatching it to the systemic tissues. Generating that pressure dictates the anatomy of the heart. The top chambers, the atria, have thin walls built just to receive blood. The bottom chambers, the ventricles, are thick and heavily muscled, especially the left ventricle, which must force blood to the furthest extremities of the body. This cardiac muscle is myogenic. It generates its own electrical impulses without waiting for instructions from the nervous system. The sequence begins at the sinoatrial node, the biological pacemaker embedded in the right atrium. The sinoatrial node fires, sending a wave of electrical depolarization across both atria. This causes them to contract simultaneously, an event called atrial systole. The electrical wave cannot cross directly into the lower chambers. Instead, it is captured by the atrioventricular node and intentionally delayed. This brief pause gives the atria time to empty their contents completely. The signal then shoots down conductive fibers called the bundle of His, straight to the bottom of the heart. Triggering the signal at the apex forces ventricular systole to occur from the bottom upwards. The ventricles wring themselves out, pushing the maximum possible volume of blood up into the exit arteries. We record this sequence using an electrocardiogram. The P wave registers the contraction of the atria. The massive QRS complex captures the high-voltage ventricular systole. The final T wave tracks the electrical recovery as the ventricles relax. Any mechanical failure usually starts as an electrical failure. Tachycardia compresses the waves, indicating a resting heart rate over 100 beats per minute. Bradycardia stretches them out below 60. In atrial fibrillation, chaotic electrical signals cause the atria to twitch randomly, destroying the P wave entirely. The heart's mechanical output relies entirely on this intricately timed electrical routing system. Once the blood is in motion, it obeys strict fluid dynamics. Blood only flows down a pressure gradient. This graph maps the pressure changes during a single heartbeat on the left side of the heart. It tracks 3 intersecting lines: the left atrium in green, the left ventricle in red, and the aorta in orange. As the atrium undergoes systole, its pressure rises slightly, pushing the final volume of blood down into the relaxed ventricle. Then the ventricle begins to contract. Look at the red line. Pressure spikes instantly. The precise millisecond that ventricular pressure exceeds atrial pressure, the fluid tries to move backwards, which slams the atrioventricular valve shut. The ventricle continues to squeeze until its pressure curve crosses above the resting pressure of the aorta. That differential physically forces the aortic semilunar valve open, ejecting blood out of the heart. During relaxation, or ventricular diastole, the red line drops. Once ventricular pressure falls below aortic pressure, the blood in the aorta tries to drain back in. It catches the pockets of the semilunar valve and snaps it shut. The valves of the heart are purely mechanical doors. They are not controlled by muscles or nerves, but are forced open and shut entirely by dynamic fluid pressure. Blood leaving the left ventricle is a surging, violent force. Delivering it requires pipes adapted to extreme conditions. The histology of an artery reflects its environment. The thick outer layer is woven with fibrous collagen for structural strength. Inside that is a dense layer of smooth muscle and elastic tissue, all wrapped around an ultra-smooth lining of endothelial cells to minimize friction. When the high-pressure pulse from the heart arrives, the elastic fibers stretch outward to absorb the shock. As the pulse passes, the elastin snaps back to its original shape. This elastic recoil maintains pressure on the blood, keeping it moving forward even when the heart is between beats. As arteries divide into smaller arterioles, the elastic tissue decreases, and the proportion of smooth muscle increases. By contracting and relaxing this muscle layer, vasoconstriction and vasodilation, the body directs exactly which organs receive blood. By the time blood reaches the veins for the return trip, it has lost its pulse and is moving under low pressure. Consequently, veins require much thinner walls with little elastic tissue, creating a wide central lumen that allows large volumes of sluggish blood to flow easily. Moving this low-pressure blood upward against gravity requires external help. The contraction of nearby skeletal muscles compresses the veins, squeezing the fluid upward. Internal one-way valves immediately snap shut to trap the blood and prevent it from falling backwards. The architecture of the piping dictates the flow. Every vessel is precisely calibrated to handle the specific pressure dynamics of the blood it carries. All of this mechanical routing exists to service the capillary bed. Here, the vessels branch extensively to maximize surface area, and their walls thin out to a single endothelial cell, keeping the diffusion distance as short as possible. This chart diagrams the microscopic forces governing exchange. Fluid movement across the capillary wall is dictated by two competing pressures. Hydrostatic pressure from the pumping heart pushes fluid outward. Simultaneously, large plasma proteins trapped in the blood create oncotic pressure, pulling water inward. At the arterial end, hydrostatic pressure is dominant. It overpowers the oncotic pull, forcing water and dissolved nutrients out through tiny gaps between the endothelial cells in a process called ultrafiltration. This escaped liquid becomes tissue fluid. It directly bathes the body's cells, delivering glucose and oxygen while simultaneously collecting dissolved carbon dioxide waste. As the blood flows toward the venous end of the capillary, the loss of fluid causes the internal hydrostatic pressure to drop. The inward oncotic pressure from the plasma proteins is now the stronger force. It osmotically pulls roughly 90% of that water back into the bloodstream. The remaining 10% of the fluid doesn't just pool in the tissues. It is absorbed by the lymphatic system, a parallel network of blind-ended vessels that slowly routes the excess fluid back into the veins. The hydration and chemical balance of every cell relies on this continuous, localized tension between physical pressure and osmotic pull. Which brings us back to hemoglobin. Our fully loaded protein has now arrived in the active capillary bed, a zone highly saturated with carbon dioxide. As carbon dioxide diffuses into the red blood cells, it reacts with water to form carbonic acid. This immediately dissociates, releasing loose hydrogen ions. To prevent the blood from turning fatally acidic, the hemoglobin protein binds to those loose hydrogen ions itself. This chart isolates the result, the Bohr effect. Binding to hydrogen ions forces the hemoglobin to alter its physical structure, causing the entire oxygen dissociation curve to physically shift to the right. Check the data point. Because of the rightward shift, the hemoglobin now has a much lower affinity for oxygen at the exact same partial pressure. It cannot hold on to its cargo. The protein actively drops its oxygen, ensuring maximum delivery exactly where carbon dioxide production proves the demand is highest. The mammalian circulatory system operates as a unified biological engine. Electrical signals dictate mechanical force. Mechanical force creates the hydraulic pressures that drive fluid exchange. And at the microscopic level, chemical gradients guarantee that the molecular payload is deposited precisely where life requires it.